Hello, this is Anika Tahsin. As you can see on the screen, this morning I'm presenting a poster on my dissertation research project, which I did while pursuing my MSc in the University of Sheffield. The project is titled as Determining the Influence of Antero-Posterior Axial Identity of the Motor Neurons on their vulnerability to the ALS predisposing mutation by transcriptome analysis. So first, I will point out some key background information and the aim of this study, and then I will talk over shortly about the methodology. And next, I will show some highlights from the results section. And finally, I will conclude with some limitations of our study as well as some suggestions for the future experiments. So, all right, let's dive right in. So ALS, that is amyotrophic lateral sclerosis, it is the third most common neurodegenerative disease, and it is characterized by progressive death of the motor neurons in the primary motor cortex, brainstem, and spinal cord. 10% of the ALS cases are inherited as an autosomal dominant pattern, where mutations in the gene for intracellular scavenging enzyme, that is superoxide dismutase 1, SOD1, are linked to the motor neuron dysfunction and in 20% of familiar ALS cases. The most affected nerve cell in ALS is the motor neurons that are arranged in the separate anatomical columns along the vertebral antero-posterior axis, that is from head to the sacrum. And during embryogenesis, ranked expression of the Hox genes designs the blueprint of the distinct neuronal fades along this craniocaudal axis. So the specific contribution of this antero-posterior positional identity of the motor neurons to their vulnerability to the ALS, it's not understood clearly still now. Some specific subpopulations of the motor neurons show more susceptibility to ALS than others. Spinal motor neurons and hypoglossal nerve that controls our tongue movement, that motor neurons, are the first group of the motor neurons to degenerate in the ALS, while the third cranial nerve, oculomotor neurons, and the motor neurons of our sacrum region, that is on-off nuclei, they escape from the deterioration even up to the end stage of the degenerative disease. Therefore, the aim of our study was to examine if the same ALS mutation affects motor neurons of distinct axial identity in the same or different manner. So to test this hypothesis, we selected a publicly accessible dataset from the GEO database of the NCBI, which contains high throughput mRNA sequencing data of the spinal cord samples with two different axial identities, that is cervical, that is neck region, and lumbar, that is the waist and the hip region. And it was collected from the transgenic mice with G37R SOD1 mutation as well as from the wild-type healthy mice. Then RNA sequence analysis was performed to observe the transcriptome alterations occurred in the two distinct groups of the spinal motor neurons from SOD1 transgenic mice when compared with those from the healthy controls. For this purpose, we used Dego's web platform for determining significantly differentially expressed genes at transcript level in the cervical and lumbar spinal cord regions of the SOD1 mice and as compared to the healthy controls. VENI 2.1 Web 2 was used to determine commonly and exclusively differentially expressed genes in the cervical and lumbar samples of the SOD1 mice. Then top gene web total was used for gene ontology enrichment analysis to identify the biological processes impacted in cervical and lumbar motor neurons of the SOD1 mice to perceive any difference between their vulnerability to the ALS owing to their axial identity. Now let's see what we have found through the transcriptome and analysis. So in the results section, in the uppermost In the uppermost part of the result section, we can see that two separate MA plots created by the biggest web tool in the left and in the right. So the left one, it is showing the transcripts with the enhanced or depleted expression in the cervical samples of the SOD1 mice in comparison to the age-matched wild-type controls, that is a healthy controls. We can see that CD5L, MCOL, L3, HTR2B, EPOC2, they are the top ranked upregulated genes, and DDX3Y, EIF2S3Y, UTI, KDM5D, NPCD are the top ranked downregulated genes in the cervical motor neurons of the SOD1 mice. If we look at the MA plot of the right side, this is displaying the transcripts with increased and decreased expression in the lumbar samples of the SOD1 mice in comparison to the healthy controls. ATP6V0, D2, MCOLN3 were found commonly upregulated. DDX3Y, EIF 2S, 3Y, UTY were found commonly downregulated in the both lumbar and cervical regions of the SOD1 mice. Conversely, GDF1, TGM1, and MMP3 were exclusively upregulated, and MYH4, TMEN233 were exclusively downregulated in the lumbar motor neurons of the SOD1 mice. Below this, on the left side, we can see that two pie charts. We can see two pie charts showing statistics of upregulated and downregulated genes. 34% of the upregulated, so the pie chart above that is showing that 54% upregulated genes in the SOD1 mice were common in both cervical and lumbar regions, and 34% of the upregulated genes showed enhanced expression exclusively in the cervical region, and the rest 12% of the upregulated genes showed enhanced expression in the lumbar region. On the other hand, higher percentage of the downregulated genes in the SOD1 mice shows depleted expression exclusively at the cervical level than in the lumbar spinal cord levels, whereas only 11% of the downregulated genes were common in both cervical and lumbar samples of the SOD1 mice. Then we can see there are four charts of the gene ontology functional enrichment analysis. The gene ontology enrichment analysis reveals the biological processes and signaling cascades related with the ALS pathology. The most enriched pathways concerned with the upregulated genes in the cervical and lumbar regions of the SOD1 mice are involved in the defense response, leukocyte activation, regulation of the immune system process, and inflammatory response, and like CD5L, ATP6V0D2, MCOLN3, these upregulated genes are implicated in the immunological and inflammatory pathways. On the other hand, the gene transcript that were found depleted commonly in the cervical and lumbar motor neurons of SOD1 mice are involved in the lipid biosynthesis and metabolism, suggesting a link between alteration in the myelin cholesterol and inexorable motor neuron death in the ALS. The exclusively downregulated genes in the mutant SOD1 cervical spinal cords are implicated mainly in the transmembrane transport and cell-cell signaling, while exclusively downregulated genes in the lumbar region, they are suggesting a link between the alterations of cell fate commitment and neural fate specification. So in conclusion, we can say that this analysis arrays a snapshot of the deregulated pathways related to the SOD1 linked ALS, uncovering that neuroinflammation is common in both the cervical and the lumbar motor neurons in the ALS model mice, but transcriptomic fluctuations are more significant at the cervical, that is in the neck region of the spinal cord, than the lumbar region, that is the hip region. So some limitations of our study were that there was lack of transcriptomic data of the thoracic, that is our chest, and the sacral spinal cord motor neurons, and unavailability of samples from the patient samples, that is from ALS patients, and about other ALS predisposing mutation, except the SOD1. Considering this study as a starting point, further experiments such as the RNA sequence analysis using large sample size dataset or transcriptomic reports on the in vitro derived motor neurons with different axial identities like thoracic or sacral samples can be performed to address the candidate biological pathways that is involved in the differential neurodegeneration that happens in this disease and in different anterior-posterior axial identity regions. Thank you for your patient hearing.